So good afternoon, everybody. Myself Kunal, and I am a junior faculty in Augusta University. And today I will talk about opportunities and danger in biotechnology. So first of all, we should know what is biotechnology. Biotechnology is the study of biology or biological materials to create a productive technology that may improve the life quality of life. It may include molecular biology, genetic engineering, and other tools that will lead us to develop the agriculture, medical science, and textile production, chemicals, and there are so many so far. So this is not a new technology. Traditionally people are practicing this technology, but we are not giving it term the biotechnology. It was at that day it was food processing or something we can say. So people were making the cheese, bread, yogurt, wine. These all are the biotechnological product. We name it biotechnology, but it was present there. The fermentation technology, people use it for change the nutrition value, to preserve the food, and to make the aroma, flavor, and texture of the food. So fermentation help in these things. So these are not a new technology. These are very old. But the area where it is applied, so in pharmaceutical company, to developing new drugs, vaccines, and many other therapies like personalized therapy. And in agriculture, improving the crop yield, developing pest-resistant crop, enhancing the food productivity. You may know the hybrid yield. So there are many technology we are using in it. Healthcare, developing diagnostic, we can know the disease earlier with the help of biotechnology, personalized medicine, as I mentioned, and gene therapies. I will come with this. And environmental science, like today the pollution is the main concern to all of us. So bioremediation to clean up the polluted land, soil, or places, pollution control, sustainable resource management, like we are producing green petroleum, like biodiesel and all. So we can produce these things and with the help of biotechnology. Food science, developing food processing techniques, enhancing safety, creating new food products. That is the areas of application. Now, it also evolved now. With the help of artificial technology, we can analyze the big biological data set, which is generated by the, you may have known about the whole genome sequencing of the multiple organism. There are lots of data, and we have to use it by artificial intelligence sometimes. Before artificial intelligence, people are using bioinformatic techniques to analyze and to discover where is the problem and how we can treat them to develop a drug. Gene editing and utilizing this technique, CRISPR. CRISPR is very known, and it is applied in research and other therapeutic purposes. I will show you one of its example in my research, what I am doing in my university. But first we need to complete this. Precise medicine, tailoring treatment and individual patient based there. Genetic makeup and other factors. Synthetic biology means there are now, we can make new biological organism which have some new characteristic that was not present in them. So that is called synthetic biology, engineering biological system to create new functionalities and products. That is, one of them is I will give you the example, the algae, which is now producing more petroleum. However, it was producing less. Now we have improved it, and it producing lot of amount of petroleum. So this is part of my research here. nowadays people are known about the colorectal cancer and IBD, means inflammatory bowel disease. So if you will see, people are taking less fiber food, and the disease is now coming more and more, means people have more colorectal disease or IBD. But these are the genes, you may know, these are gene which is expressed on the cell surface in the colon. So, however, it is expressed means all over the body, but majorly in the colon, they absorb the fermented product of the colon. Bacteria, if we are taking high fiber, bacteria degrade them, they break it, and they make the butyrate and short chain fatty acids. And these fatty acids are actually very important for our health. They, when it is absorbed, it comes to our system and it reduces the inflammation and other things. And it will prevent the colorectal cancer and IBD. So here what we did in the case of our research, sorry, it is not pointing, yeah. It is not showing that, sorry. So we have taken this GPR 109A. These are actually the receptor which is responsible to transport the short chain fatty acid and butyrate from the colon to the inner side of the body. So we have selected two particular genes like GPR109A. However, these genes are also involved in this, and these are the transporter, direct transporter. So they also take actively, means with the help of energy, they are also transporting those materials towards the cell. So we have, using that CRISPR-Cas technology, to delete these two genes from the mouse, and we develop a mouse which have not expressing these two genes in their body. So then we checked how it is responsible or not, or what is its effect on the immune cells. So you can see here, I'm sorry, it is not working that way. So here you can see, so this is GPR109 knocked out mice, and here SLC5A, which is also called SMCT. So this is the both gene which is knocked out in this mice. It is GPR109 knockout, here it is SLC5A knockout, and here it is both in the same mice knocked out. So what we see, the FOXP3, which is the immune suppressor, in the case of, and if immune is not suppressed, it will cause the inflammation sometimes in the case of bowel disease. So here, you will see when we knocked out, there as FOXP3 expressing CD4 cells count was very down. So that means this gene is responsible to produce the FOXP3. And here also, it is the same way, because this is the transporter, this is the receptor. And if you will see the anti-inflammatory cytokine secretion, interleukin 10, their secretion goes down. So, and here you can see both of the case, it is down. And pro-inflammatory, which is responsible to induce the inflammation, their expression, when we knocked out these two, their expression went up. So that indicates, so these receptors are responsible, and they have a major role in the colorectal cancer. However, our research, this is the part of research, and we have to work on very long on it. Then we will make the bigger conclusion on it. But this is the preliminary result. And that's how we can apply the technology, CRISPR-Cas and other technology, to improve the research and developing a new therapeutics. So now I will come to the opportunities in biotechnology. So healthcare innovation, personalized medicine, and genomics, you may see there are different persons and they have different types of However, disease is same, but their reactivity with the drug is different. So we can develop a personalized therapeutics, early diagnostic. You can check it with the, there are many techniques like PCR and all real-time PCR we can use to detect and diagnose, early detect the disease. So regenerative medicine nowadays very much in the fashion. You may have heard about the CAR T cells therapy and stem cell therapy. These are all the regenerative medicine, and CAR T cell therapy also are one of them. And where you may we can take the T cell from the patient and we transform it with the new receptor, and then we again inject to the patient. That will now kill the disease causing cells or diseased cells. So that is the CAR T cell therapy, which is again the product of biotechnology. Now, the agriculture sustainability. So here, climate resilient crop, reduced pesticide use. This is common thing, you know. Nutrient enriched food like golden rice, people are starving because of this problem. So because they don't have vitamins, so now we have incorporated that gene which is present in the rice. If you will eat rice, you will get the vitamin A. So now environmental solutions, microbial oil spill cleanup. This is the bigger issue nowadays for environmental concern, because if you will spillage of the oil in the ocean, in the river, that kills many aquatic organisms. So if we will clean up with then it will be a good, healthier environment. So there are nowadays pseudomonas species of bacteria which is responsible to clean up all the hydrocarbon-related pollution. Enzyme-based plastic degradation, you can see now there are bacteria available which is now degrading, which is capable to degrade the plastics. And carbon captured with green engineered algae, like here you will see the algae is responsible to, they are taking carbon dioxide, and they are sequestering the carbon dioxide in them, and then producing the biofuel and other livestock, fertilizers and other things. Economic opportunity, global biotech market growth is very much promising. You can see it is growing. And job creation in multiple sectors. This is giving, we are also working in that way. So, and it also developed a biotech hub, Boston, San Diego, Singapore. These are the places, but it also brings some dangers with it. So what are the dangers? Potential misuse in bioterrorism. So we can do the genetic manipulation in bacteria to get the beneficial thing. At the same time, we can also incorporate that gene which can cause severe damage and kill several people at a time. So that is again the bad side of this. Accidental release and harmful organism, you can see that can happen with this. And that's why it's a dilemma, whether we should pursue this or not. So that is the risk of biotechnology. Again, ethical concern, germline, editing, and designer babies. Now, because this is possible, we can do any type genetic manipulation even in the early stage of the embryo development. So we can incorporate our desired genes. Sometimes we can ask, yes, this famous actor, I need their gene in this. This scientist, I want this one in this, and I want their eyes like this. So we can make a designer baby. That is again the major concern. And we can also lose our genetic code privacy. If I have done our single-cell RNA sequencing or our whole genome sequencing, and if it is still out, there will be a problem. So here you can see if there will be a designer baby. And socioeconomic equality in access. Yes, this is major concern. The people who have money, they can access it easily. the people who don't have money, they will not grab this technology and they will be poor still. So this is the, and last, this environmental risk. Gene flow in wild type species. Suppose we have changed some genetic makeup in an organism and it goes to the environment and it mixes up with the native flora and fauna. So that will disturb total ecosystem. So similar way, ecosystem imbalance, if we will suppose modify this mouse or any organism out of this, these are the pests you may know. So if we, to prevent them, we can modify them. If this will be not present, this will not survive. Again, this will not survive, and its whole ecosystem will be collapsed. Irreversible synthetic biological effect, again, that is the same way. If we have manipulated any microorganism, and that is because we have developed them resistant thing, everything, means suppose disease resistant microorganism or drug resistance, ampicillin and antibiotic resistant. Suppose we develop a bacteria, and because it is already resistant, so how we will kill them again, if we have to kill them? So it will again make a bigger problem. And now the regulatory gaps. So there are global differences in biotechnological laws. Many countries allowing it, many countries not allowing. Somebody is saying yes, somehow no, and some way, okay, somehow we will accept it. So yes, this is the situation. Somewhere you will go yes or no, or maybe regulation lags behind the innovation. Again, if you will have problem, or we have much regulatory strict restrictions, then it will again stop the research growth in a positive way, not in a negative way. Past regulatory failure means, suppose if we pass the regulation, okay, this is allowed to do. Again, after investing lots of money, there is a chance we can be failed at the end. So these are the dangers, and how we will do this, balancing benefits and risks. So responsible research and innovation, we need to take care of all those things. We have to be very much careful handling the technology. Global cooperation is also needed, and public engagement and transparency is very important in this one. So on the conclusion, opportunity, healthcare, agriculture, environment, economy, and danger, biosecurity, ethics, environmental risk, regulation, and the balanced one is informed decision making. So that's all for today's presentation. Thank you. If you have any question.
Does anyone have any questions? No questions?
Yes.
Thank you, Dr.
Thank you.